Protein structure. The function of a protein can only be understood in terms of its structure. Protein structure is discussed in terms of 4 levels of organization. Primary structure is the amino acid sequence of its polypeptide chains. Every protein has a unique amino acid sequence. Secondary structure is spatial arrangement of the polypeptide backbone, ignoring the conformation of the side chains. Tertiary structure is a three-dimensional structure of the entire polypeptide. Quaternary structure refers to the three-dimensional structure of proteins that are composed of two or more polypeptide chains called subunits. The primary structure of protein is held together by covalent bonds. Peptide bonds and three-dimensional structure of a protein may be held together by a variety of bonds like hydrogen bonds, ionic bonds, covalent bonds, and hydrophobic interactions. So first, primary structure. Each amino acid in a protein is unique, and determination of the amino acid sequence is an important part of characterizing proteins. Most protein amino acid sequences are deduced from the sequence of its gene because sequencing of DNA is much easier than sequencing a protein. However, determination of protein sequence is still an important tool in biochemistry. The primary structure of a protein comprises of the sequence of amino acid linked via peptide bond. The amino acid sequence refers to the sequential order of a polypeptide. The relative amount of each of the 20 amino acid is in a peptide is referred to as the composition of the protein. Amino acids are joined together by the peptide bond that is formed by the reaction of the alpha carboxyl group of one amino acid with the alpha amino group of the another amino acid. If the process is repeated many times, then a long linear chain of amino acids, polypeptide, is produced. Repeating polypeptide give rise to peptones, proteoses, and proteins. Peptones and proteoses are small molecules formed of a few amino acids. Primary structure represents the simplest level of protein structure. The sequence of the different amino acids is considered the primary structure of the peptide or protein. Still, it represents the functional role of a protein. Each amino acid in a protein is unique, and determination of the amino acid sequence is an important part of characterizing proteins. Most amino acid sequences are reduced from the sequence of genes because sequencing DNA is much easier than sequencing of proteins. However, determination of protein sequence is still an important tool in biochemistry. So, 4 levels of protein structure can be recognized: primary, secondary, tertiary, and quaternary. Primary structure protein is preserved by end-to-end covalent peptide bonding between amino acid residues, and others by non-covalent bonds or forces such as interpeptide hydrogen bonds, side chain hydrogen bonds, disulfide bonds, ionic bonds, apolar or hydrophobic interactions, van der Waals forces, etc., which we have already mentioned. Then the primary structure of proteins, that is nothing else but just the sequence of amino acids. Primary protein structure is the amino acid sequence or the sequential covalent linkage of amino acids in a linear polypeptide chain. So it is also called polypeptide chain structure of amino acid sequencing structure. Examples are insulin, glucagon, etc. The amino acid sequence in a peptide chain is conventionally read on the N-terminal to C-terminal direction. This is typically analogous to the primary structure of nucleic acids, nucleotide sequence. Thus, there is a strict collinearity between the N to C polarity amino acid sequence of polypeptide chain and the 5 to 3 polarity nucleotide sequence of its cognate mRNA. Primary protein structure is specified or determined by the nucleotide sequence or genetic code of the DNA that governs protein synthesis. It is established by genetic transcription and translation. This primary protein structure serves as the fundamental link between genetic code and physiological function. It is the primary structure of a protein which determines their physiological role. In primary protein structure, amino acids undergo condensation polymerization through covalent peptide bonding. Covalent peptide bonding, amide linkage, to form unbranched linear polypeptide chains. Polypeptide linkage is incapable of releasing or accepting protons, hence it does not continue to the acid-base properties of polypeptides. What is that? Peptide linkage is incapable of releasing or accepting protons, hence it does not contribute to the acid-base properties of polypeptides. Peptide bonding occurs between the C of the COOH of the one amino acid and the N of the H2 group of another with the elimination of one molecule of H2O. This H2O is formed from the OH of the COOH group and the H of the NH2 group. Consequently, the linkage between C double bond O and H, this is peptide bonding. Here O is a slight negative charge and H has a slight positive charge. Therefore, each of them can participate in the formation of hydrogen bond. This hydrogen bond capability is important in determining the structure of a protein. Protein bonding is an endergonic process. That means hydrolysis of peptide bond is hexagonic, but this is an endergonic process. It requires an input of energy and a series of reactions catalyzed by the enzyme peptide synthetase. It occurs on polysomes or ribosomes. Once the amino acids are peptide bonded, they are called amino acid residues. They are no longer amino acids because they have lost a hydrogen atom from the amino group and a hydroxyl moiety from the carboxyl end. A peptide chain formed of two amino acid residues is known as dipeptide. That which consists of a few amino acid residues is called oligopeptide, and that which contains many amino acid residues is called a polypeptide. Repeating polypeptides give rise to peptones, proteoses, and proteins. Peptones and proteoses are small molecules formed of a few amino acid residues. The term protein is usually used when the molecular weight exceeds 5000. Some proteins consist of only a single folded polypeptide chain. Others consist of two or more identical or non-identical chains held together by non-covalent bonds. A polypeptide has a main chain and several variable side chains. Main chain forms its backbone. It has two distinct ends, namely the alpha amino terminal and the alpha carboxyl terminal or C terminal. The former bears a free terminal alpha amino group and the later has a free terminal alpha carboxyl group. The N terminal is taken as the initial end, so N to C polarity will be there. In a polypeptide, only the free terminal alpha amino and alpha carboxyl groups are ionizable. The others will not ionize since they are covalently joined together through peptide bonds, so they do not contribute to the total acid-base behavior of peptides. In proteins, hydrophobic amino acids prefer to be located near the center of the molecule, and hydrophilic amino acids prefer the outer side. This peculiar orientation of amino acids is achieved by the characteristic folding of the polypeptide chain. Primary structure represents the coolest level of protein structure. Still, it determines the functional role of a protein. Its stability is achieved by the covalent peptide bonding between the alpha carboxyl and alpha amino group and adjacent amino acids. It is significant in having a unique and genetically determined amino acid sequence. Next, let's learn about the secondary structure of protein. If the peptide bonds were the only type of linkage present in proteins, these molecules would have behaved as irregularly coiled peptide chains of considerable length. But the globular proteins, however, do show some regular characteristic properties indicating the presence of a regular coiled structure in these molecules. This involves the folding of the chain, which is mainly due to the presence of hydrogen bonds. This folding and hydrogen bonding between neighboring amino acids results in the formation of a rigid and tubular structure called a helix. This constitutes the secondary structure of proteins, which refers to the steric or spatial relationship of amino acids that are near to each other in the amino acid sequence. Secondary structure determines the steric or spatial arrangement. Based on the nature of hydrogen bonding, Pulling and Corey identified two regular types of secondary structure in proteins, alpha helix and beta pleated sheet. So, secondary protein structure represents the ordering of the otherwise extended peptide chains to make the protein molecule short, rigid, and compact. It is accomplished by intramolecular hydrogen bonding and other weak bonds between the carboxyl oxygen and the amino acids of the polypeptide backbone. This is found in fibrous proteins such as keratin of hair, collagen and elastin of connective tissue, myosin of muscles, fibrin of blood clot, fibroin of silk, etc. Very rarely polypeptide chains remain fully extended. In most cases, one part of a chain sticks to another part through hydrogen bonding. This intra-chain hydrogen bonding and the periodic spacing of peptide bond units result in a variety of molecular conformations. This broad classes of secondary protein structures can be recognized, namely helices, sheets, and turns. Of this, alpha helix structure and beta plated structure are the commonest ones. Secondary structure not fitting for any of the three major categories are described as coils. So, which are the three types of secondary structures? Helix, sheets, and turns along with coils. Okay. So, first let's learn about the alpha helix structure. This is the secondary protein structure in which a polypeptide chain undergoes right-handed helical coiling and intra-chain hydrogen bonding between successive coils. It forms a stable right-handed spiral spring called alpha helix, which is stabilized by numerous hydrogen bonds between CO and NH groups. Left-handed alpha helix is rare because amino acids of proteins are L isomers, which exclude left-handedness. Amphipathic alpha helixes will have nonpolar hydrophobic residues on one surface and charged or polar and hydrophilic residues on the opposite surface. This enables them to interact with both polar and nonpolar chemical groups. In the alpha helix, hydrogen bonds are formed between the carbonyl oxygen and imide hydrogen along the polypeptide backbone. So the C and N atoms of the peptide bond participate in hydrogen bonding. But the R groups of this are project outward from the helix. Hydrogen bonds are almost parallel to the axis of the helix. Usually, alpha helical configuration spans over 10 to 15 amino acid residues. It would be then interrupted by non-helical stretches. Keratin, hemoglobin, myoglobin, etc. are proteins with high proportion of alpha helix structure in their polypeptide. H bonds are vertically shown, side chains will be horizontal. That is the adjacent folds hydrogen bonding. Alpha helix, the right-handed helix, is the most common arrangement. Alpha helix results from the conformation of a peptide bond and R group seeking the most stable arrangement. There are 3.6 residues per turn in right-handed helix. The periodically or distance that the helix rises per turn is 0.54 nm. The rise per amino acid is 0.15 nm. H bonding between every fourth peptide bond stabilize alpha helix in helical region of the protein. Every peptide bond therefore participate in H bonding, which leads to a very stable structure. A consequence of the alpha helix is that amino acids apart in primary structure are spatially close to each other in the helix. In helical region of a protein, every peptide bond therefore participate in H bonding, which leads to a very stable structure. A consequence of the alpha helix is that amino acids apart in primary structure are spatially close to each other in the helix. Hydrogen bonding is the strongest when groups are in straight line, which is the configuration of alpha helix. Tight pack within the core of the helix also stabilizes the helix. The alpha helix is a rod-like structure. The tightly coiled polypeptide chain forms the inner part of the rod and the side chains extend outward in a helical array. The alpha helix is stabilized by hydrogen bonds between the NH and CO groups of the main chain. The CO group of each amino acid is hydrogen bonded to the NH group of the amino acid that is situated 4 residues ahead in the linear sequence. Thus, all the main chain CO and NH groups are hydrogen bonded. Next is the beta pleated sheet. Beta strands are typically 5 to 8 amino acid residues that form an extended chain of silk spider webs. They form a fully extended structure. Adjacent amino acids are 3.5 angstroms away. These strands can form sheets when two strands are grouped and hydrogen bonded. Arrangements of beta sheets can be antiparallel, which have amino terminal end at one end of the polypeptide oriented towards carboxy terminus of polypeptide. Antiparallel is the most common arrangement. Parallel, which have amino and carboxy termini of peptide chains, are oriented in the same direction. They are often grouped in a structural units of 2 to 5 sheets. Beta sheets can have a mix of parallel and antiparallel components. This is a protein structure in which a polypeptide chain folds back upon itself in such a way that two parts of it lie side by side in parallel or anti-parallel arrangement. This permits hydrogen bonding between adjacent parts of the same chain. In parallel arrangement, two regions of the polypeptide chain lie side by side with their amino terminals pointing in the same direction. In parallel arrangement, two regions of the polypeptide chain lie side by side with their amino terminals pointing in the same direction, beta keratin. But in anti-parallel arrangement, two regions of the chain lie side by side with their amino terminals pointing in opposite direction, for example fibroin. In a beta pleated sheet, the R groups project above and below the plane of the sheet. Beta sheets are usually twisted in a right-hand direction, sometimes to such an extent that they form parallel-like structures, propeller-like structures. Beta pleated structure is characteristic of the proteins which make silk fibers. The sheen of silk is due to the reflective property of the beta pleated sheets. Tertiary structure conformation of polypeptides. This is the extensive helical twisting and irregular folding, bending, and looping of the polypeptide chain. It makes the protein molecule compact and almost spherical. It is characterized by the covalent interaction between the side chain radicals of the different amino acid residues and also between side chains and amide groups. Tertiary protein structure is stabilized chiefly by disulfide bonds, which are the only covalent linkage involved in tertiary protein structure. What is the main and one and only chemicals bond involved in the tertiary structure? That is nothing else but the disulfide bonds. Non-covalent interactions include polar bonds, hydrogen bonds and ionic bonds, and non-polar bonds such as van der Waals interactions. Tertiary protein structure is energetically most stable and is characteristically found in most biologically active proteins. It is important in that biological properties such as enzyme activity, antigenicity are related to it. In tertiary protein structure, R groups have a significant role. Through this interaction with each other and also with water, they influence the folding of the polypeptide chain. Hydrophobic R groups interact with one another due to their non-interaction with water. As a result, the polypeptide chain tends to fold in such a way that the hydrophobic R groups are concentrated inside the fold without contact with water. At the same time, the hydrophilic R groups remain localized at the surface for maximum polar interaction with water. Thus, in tertiary structure, a balance is established between the internalization of hydrophobic R groups and the localized externalization of hydrophilic R groups. Tertiary structure represents spatial arrangement of amino acids that are far apart in the linear sequence. It results from interaction between R groups via van der Waals sources ionic hydrophobic and hydrogen bonding and disulfide bonding. Grouping or arrangement of secondary structure into combination that are present in a variety of proteins are often described as motifs or domains. Examples are parallel and antiparallel beta sheets. Greek key motif 4 adjacent antiparallel beta strands, beta alpha beta motif. That means can be either 2 parallel or anti-parallel beta. All this. Then the quaternary structure. Many large proteins contains more than one polypeptide chain. The spatial arrangement of the subunits is the quaternary structure of the protein. The forces that hold subunits together are some weak bonds that as those that stabilize the tertiary structure of proteins, van der Waals and London dispersion forces, salt bridges and hydrogen bonds. The contact region between subunits resembles the interior of a protein. The subunit may be identical, but there is always defined stoichiometry. So, quaternary structure is the self-assembly. Polypeptides get together. This is the interaction, orientation, and organization of two or more polypeptide chains through weak non-covalent bonds such as hydrogen bonding, ionic bonds, hydrophobic interactions, and van der Waals forces to form a multi-stranded or multi-unit functional protein. Primary, secondary, and tertiary structures involve only a single polypeptide chain, but quaternary structure involves more than one chain. The constituent polypeptides of a multi-stranded protein are called subunits or monomers. A multi-stranded protein with different kinds of subunits is called a multimer or heterogeneous proteins, hemoglobin. And the one with at least two identical subunits is called oligomer. The identical subunits of an oligomer are called protomers. Protomers are bound together by weak non-covalent interactions. Based on the number of subunits, multi-stranded proteins can be classified into dimers, two subunits, tetramers, hexamers, and polymers. Quaternary structure is typically found in hemoglobin, immunoglobulins, many enzymes. Creatine phosphatase is a dimer, lactic acid dehydrogenase is a tetramer, aspartic acid carbamylase is a hexamer. A hemoglobin molecule is a tetramer with two pairs of protomers, each of which is formed of two peptide chains. Its protomers are two identical alpha chains and two identical beta chains. An antibody molecule or immunoglobulin is also a tetramer formed of two identical heavy chains and two identical light chains.